of genetics can be traced back to ancient times. Humans have practiced genetic manipulation long before the discovery of DNA. Since ancient times, animals and plants have been bred for specific uses and intent. Such selective breeding practices were the forerunners of modern science of genetics. Then came James Watson and Francis Crick with their discovery of DNA in 1953. This transformed the way we think about diseases and ailments that are primarily caused by genetic mutations. In an attempt to treat or even cure these diseases, scientists came up with a concept called gene therapy. To better understand how gene therapy works, let me first explain how DNAs are expressed into proteins. DNAs are the chemical building blocks of life. They form chains of sequences that are first transcribed into RNA and then translated to form proteins that are essential for life. Each DNA sequence that contains instructions to make a protein is known as a gene. A genetic disorder is an ailment caused by mutations in a gene. These mutations can be thought of as a change in DNA sequence. This mutation results in defective genes being expressed into dysfunctional proteins, thus giving rise to the disease. This is where gene therapy comes in. Here's a short explanation of how gene therapy works. The target cells are first removed from the patient. In the laboratory, viral genetic material is rid of its original pathogenic ability and is armed with the functional gene. The altered virus is then mixed with cells from the patient. The cells from the patient become genetically altered. The altered cells are injected into the patient. Finally, the genetically altered cells produces the desired protein. In essence, gene therapy involves substituting the defective gene in a cell with a functional one. So, what's the big deal about gene therapy? Now, the potential of gene therapy is huge. Many genetic disorders, such as congenital blindness, type 1 diabetes, and adenosine deaminase severe combined immunodeficiency, also known as ADA-SCID or Barber Boy disease, can possibly be cured with gene therapy. As a matter of fact, there are already successful gene therapy clinical trials on these genetic disorders. This could dramatically improve the life of the affected and gives people with genetic disorder a chance to lead a normal life. However, there are still controversies surrounding this seemingly miraculous medical breakthrough. The death of Jesse Gelsinger in 1999 after being administered with gene therapy highlights this controversy. This is the first major setback of gene therapy, and this got people asking whether gene therapy is truly safe. Drawbacks of gene therapy include chance of inducing tumors after being administered gene therapy, the short-lived nature of gene therapy, severe immune response to gene therapy, and possible ethical concerns. But as recent history has shown, researchers continue to make new strides in the field of gene therapy. Genetic diseases that would have been fatal can now be treated, or could potentially be cured with gene therapy. There are no doubts about the vast benefits that gene therapy can bring about, as it can potentially eradicate human genetic diseases in the future. Many of the shortcomings of gene therapy are due to the fact that the technology is still in its infancy. For these reasons, research directed at the development of gene therapy should be strongly encouraged.